Good evening, one and all. Thank you so much for joining the session. And today we will be more focusing on AutoDock. And of course, I know that everybody is using it already, but I wanted to see whether uh, you are able to get into deeper details and also able to customize if there is something that you have any glitches when you're working on. So today I will be giving you initially a demo on a case study, and later you can ask me questions if there is any specific problems. Unfortunately, I won't be able to take your PDB and do it for today's online live session, but I'll be happy to answer your questions after that. Of course, you can contact me through phone or through WhatsApp, whatever you prefer. If you're sending email, kindly send the details completely so that I'll be able to help you. So, as usual, if you want to ask any questions or anything, you can put it in mentee.com. But today's, I would prefer more to be on an audio discussion because rather than reading out the question, it is better to have understanding the question and moving forward. So, as usual, participants who have joined via Meet, I would recommend that you probably should follow the commands that I'm going to tell you now. Just a minute. Yes. So everybody should requested to switch off your webcam, and also please mute yourself so that we will have a good session without much audio noise backgrounds. And kindly tell your name before engaging in any discussions via audio and questions. In this case today, I prefer to have a direct questionnaire through audio, and we will decide on what is that you all know. So if you can let me know. Okay, already a few of you have already put it. So if you could let me know what is your interest today, whether it is target definition, result analysis, mainly on AutoDock or AutoDock MGL tools or AutoDock tools, or even AutoDock Vina, or file formats and conversions, or the basics of whatever you see in the tutorial. So you can go to mentee.com in a web browser and give the command 650379 so that you will be able to choose which one you actually wanted to today concentrate on. So if you need some more time, I'll wait for a few more seconds. I can see already 30 people have already put their casting their vote. I can see more of target definition and also on result analysis is what you are more looking at. But if there are more people who want to see some others, of course you are happy to put in there. Yeah, people are still submitting it. Okay, result analysis is what you're looking more. Okay. Yeah, so we have an overview here. So today I will, of course, more ultimately I was planning to more focus on target definition as well as on result analysis. This is very, very important. Yes, because how to do docking and all, it's already being given in many tutorials. So it's plenty of them in the YouTube. So of course, when you're doing a session, we should do something which is not right now available in the web. So to just understand the audience, could you please tell me what is your level of understanding different file formats details? I mean the details in AutoDock, and why do we generate grid files? What is necessary there? And what not to do in AutoDock, as well as kindly do not present. I would recommend do not present your screen. Um, Thank you so much for that. Let me just go back because the person has. It's okay. It happens. But by the time you can also cast your vote. So let me resume my presentation. Okay, I hope you are able to see now. Great. So this is just to understand where we have to concentrate. Now, installation of the software, either Linux or Windows or Mac, then result analysis, generation of 2D depiction, that is ligand protein interaction. This is very, very important because just putting a 3D picture on a paper or on a report, of course, it gives you some information, but most of the time, my personal choice, and I hope most of you also agree, the 2D depiction also helps you to understand a clear definition of which atom of the ligand is interacting with which atom of which amino acid, and what kind of interaction, whether it is a hydrogen bond or a hydrophobic region. So those things you can understand from 2D depiction. Of course, today we will be discussing about that also. Okay, great. I can see that what not to do in Autodoc, many of you are not aware of. But file formats, it's like a medium. So anyway, we will go through that today. Now, just an overview. Today we will be discussing about the topics that rank top, that is target definition and more of result analysis. But I'll just go through the other parts in a normal way. And all of you should refer to only open source. That's what today we are doing, open source data. And no promotions are entertained here. All the opinions and discussions are of personal view and with no connection or relation to current employers or corporates or any other individuals. I wanted to admit this because many of your participants, as well as me, working for different employers. So we want to take this disclaimer before we start. So great. Now, before going to the questions, now I will be just starting presenting now from a different machine to just to show you, a better software visibility. Um, okay, so just wanted to confirm whether you are able to. Just a minute. Okay, just let me admit them and then we will go ahead. Sorry about this. Something is popping up. Anyway, okay, so things are clear. Oh, sorry. Okay, clear. So I wanted to ensure now whether you are able to see my screen where there is a web page came with a PDB ID. So just to
Yes,
thank
sir.
you,
Yes, sir.
thank you. So in order to go in this particular anchor,
No, sir, I didn't get.
can you click on the Research India name so that you will be able to see the screen? I mean the shared screen. Probably you're on a
Sir, I got.
yes, great. Probably you might be on a different. Uh, so I would recommend you to please switch off your webcam for a better visibility.
Yeah. Yes, sir.
Yeah. Okay, great. Um, okay, so there are a few comments already there. Sir, brief about protein docking also. Unfortunately, see in AutoDock, I do not recommend to go for a protein docking because atom type definitions will be huge on the ligand side. But protein docking, we can cover it using a different tool, maybe on a later date. We can do that. Then structural approaches of docking, basics, of course, we can cover that. But also other details they wanted to interest in, we can do that. Anyway, okay, these were the usual comments that I got from everybody. So, but let me check if there's any other comments. No, everything is good. Great. So we will start then. So before choosing your target protein, of course, yesterday there was a session on polypharmacology based on molecule to target identification. So if you have any idea that you wanted to understand the definite PDB or the protein target, you need to have a better understanding there. So it is not that earlier many people worked on a particular protein, so I will also pick it up. there should be a reason behind why you're actually choosing a protein. It could be the right protein target for you, or it is bioavailable. That means it can be present in animals, or it can be, if it is in a human protein, it can be induced. So the rule of thumb is that you need to get a good, better resolution crystallographic structure, extra crystallographic structure, as well as you have to ensure that this is bioavailable, as well as the protein is druggable. If you are not able to check this, I would recommend please do not do any docking studies on it, because docking studies is for us to scale up to wet lab analysis. So we need to have a comparative analysis, whatever you are doing in silico, that can be scaled up to in vivo or as well as in vitro. You might not get the exact proteins target there on three criteria, but you should be able to be comparable, and you should be able to understand what is the significance of doing in silico studies when you're trying to do in vitro or in vivo. So this is very important. After considering that, if you're trying to search for a particular target, there might be plenty of PDBs available. Now you need to have a better understanding which PDB I have to choose. Of course, you have to read a lot of papers, try to understand which region of your protein or of the genome this is expressed as the protein region, what happens if it is getting inhibited. So we need to have a thorough understanding of the target before choosing the PDB. And after choosing the PDB, we need to also know what is our final objective. It is not docking and getting some numbers. It's about what is next. I got something on docking studies, what I'm going to do with that. Of course, that is just a hit molecule. Don't think that by just doing a docking, you can find a drug molecule or even a what is a drug-like molecule. Sorry, I have to tell you the fact, we are just finding the hit molecule when you're doing a virtual screening. But if you already know something is already acting, that is a different scenario. But when you're doing a virtual screening, we are just finding a hit molecule from our docking analysis. After finding the hit molecule, of course, there are several analyses that has to be carried out. So when you're finding out hit molecule, I'm not ending up with one single molecule. I might end up with 20 molecules or 50 molecules, because my hit
So your voice was not audible earlier.
Oh, sorry.
Yeah,
Am I audible now?
yeah, now you're audible.
Okay, thank you. So today we will take this 1DWB, which is the resolution of the binding to human thrombin of four active site directed inhibitors. So we will take this as an example, which is a hydrolase inhibitor. If I go below, I can see that this has three chains. The chain L, which is alpha thrombin, then chain H is again a larger subunit, and then chain H has a ligand molecule which is called MID. It is a naphthalene sulfonyl glycyl carbamidol um, piperidine compound. And then we have also a I chain. Now, why I picked up this, there are several references in binding DB for this particular co-crystal ligand in nanomolar scale. So I thought to choose this as a best example to do today's AutoDock exercise. Now, everybody knows that we have to download the PDB file from here, but just going to download a PDB format. But before doing that, I want to do a little bit of analysis to understand my protein a bit deeper. For that, what I do is I go to the sequence tab. Of course, you know that you have the FASTA sequence can be downloaded here. Download FASTA sequence, as you can see here. So I'm just going to download a FASTA sequence. I'm going to visualize. You can see here, this is the FASTA sequence that you can see here. It starts with a greater than symbol. The first line should be a narration of that particular sequence. It can be a DNA nucleotide or protein sequence. And from the next line, the sequence starts. Ensure that there is no space, no numbers. It is just only the single letter code of either amino acids or nucleotides. So that is called a FASTA format. Okay. So by copying that, I'm going to a website called as ProSite. So ProSite is a database of protein domains, families, and functional site. I wanted to understand a little bit more about what is motives or functional site or active site. which is being reported for this particular protein. I can just paste it here. I'm sorry. I can just paste it here. I just copy and then go and paste it here. And then I can click on scan. I'm not doing that. It might take few seconds, but I have already done that. So if you can see here, if you're not able to see, I can a little bit zoom more. I hope now it is better. I just zoomed it. Okay. So you can see there are 259 amino acids are there. In that there are 3 hits. The legends, it's already being shown. There are active sites and others. So you can see there's a trypsin domain at particular region. And you can see from 1 to 255, that is a trypsin domain, that is serine protease or trypsin domain profile. And you can see there are active site in the region of 43rd amino acid and 99. They are a charge relay system anyway. There are disulfides also in the particular residues of 173 to 187. And there's another active site on 205. And there are other active sites also. As I told you, 39 to 44, it is an active site. It is a serine proteases. And another active site, which is a predictive feature, it is from 199 to 210 with a confidence level of 0. So this is the signatures that you can see for serine protease. This is a level of understanding from ProSite when you try to take the sequence analysis. Let's go to the next part. We wanted to understand the pocket, the binding pocket available in this particular 1D WD. Let it be any PDB, okay? Now I went to a server called as CASP server. Why I recommend this CASP server? Because if you're submitting a new PDB ID there, they try to do analysis, but after that, if a second person comes and gives the same PDB ID, it loads much faster. So if you have a very low internet, this is the best one. If already someone has already done that. So here we are seeing that the surface area of the particular pocket is so much, and the volume is 286. So it's kind of a water fill algorithm. They try to do something similar to that, but it is more of a surface topology of proteins. And here at the bottom, you will be able to see which are those amino acids and their sequential ID. 48th amino acid, serine, 49th amino acid, 51st amino acid, 114th amino acid. Like that, you will get to know. Of course, you have to manually note it down. Next part is when you go to your PDB, this one, and of course, there is something even I was also, when I was starting doing all this activities, I never go to read this paper. But later I understood if we don't read that paper, you really don't understand things. So we have to read the paper completely for that PDB to understand what is the significance of the ligand, what is the significance of specific single atoms in that protein, what are the different cofactors available like NAD, FAD or heme, whatever it is. So we need to understand only by reading this. So for me, before choosing any protein for doing docking analysis, you have to go through the literature. You have to have an understanding about the functional side, that is through ProSite, and then also about the size and the amino acids that is there on a structural perspective of where the pockets are possible in this particular the PDB. After having a thorough understanding of this, of course, you structure is good. You need not to minimize the structure until if there is missing residues in your protein structure. So there are possibilities. Bilquis, if you can stop the sharing, it would be great. Let me try to share it again. Sorry about that. Okay, ensure that you don't click on the present now. Yeah. So I hope you're able to see now. Okay, great. So ensure that you are starting with a quality structure, the best quality available from an X-ray structure. So you need not to minimize it. But in certain algorithms, certain docking algorithms, there is mandatory thing that you have to minimize or apply force field. That is depending upon different algorithms. But here you have to understand, is there any missing residues of this particular protein when it got crystallized? There are chances. If there are missing residues, of course, you can use modeler or a Rosetta in order to add those missing residues. And during that time, you have to minimize the structure. It all depends upon, that's what I said. You have to have a thorough analysis before choosing which is the right protein going for docking. Anyway, so this is all with respect to the protein target. Now, next, let's go to the software side. In order to use a GUI version of AutoDock, AutoDock does not have any graphical user interface. So it is better that you go to MGL Tools. Of course, they are going to phase out this particular page soon. So you have to go to their new website, CCSB. I'm not going there now. You can do it later. So if you go to MGL Tools, you can go to on a left-hand side downloads. In the downloads, you have for Windows, for Linux, 32 and 64-bit, as well as for Mac OS. Mac OS, there is an issue. If your Mac is being updated to Catalina, that is the latest version, these won't work. So you have to wait for an update that is coming from MGF Tools. So otherwise, it's a direct install that you can do. And one more thing I wanted to refer, if you are using any Linux, already in my page in the channel, you have a video which says Linux for researchers and computational chemists. Of course, it can be for people who are doing computational drug designing also. So it's there in my the channel in YouTube. If you go to 24th minute, you will be able to see how to install AutoDock and AutoDock tools. Here I have used Linux Mint, not Ubuntu. Ubuntu, it's also similar to Ubuntu, but why I choose Mint, there are many more tools by default you will be able to install as a beginner. So that is very helpful when compared to uh, the Ubuntu platform. But if you are trying to do it on Windows, it is a straightforward. You just install the MGL tools in order to install the AutoDock tools. And then you have to go to another website for AutoDock again, that is autodock.scripts.edu, and then go to downloads, and you have to download AutoDock 4.2 as well as AutoDock Vina. Now, keep in mind AutoDock and AutoDock Vina. AutoDock Vina has a different search algorithm, which is much faster than AutoDock and can be used for virtual screening. And the parameterization is very, very minimal when compared to AutoDock 4.2. We will come across. Pyrex is a software that is being used for virtual screening. So you can load many ligands there, and at a single shot, you can run the docking. But I have experienced that Pyrex in some operating system, when you have more than 1,000 molecules, it might crash. So ensure that you have a good resources like RAM and good CPU, and don't disturb the machine when you're using Pyrex for doing AutoDock Vina virtual screening. And after that, I would also highly recommend to have OpenBabel. OpenBabel is something very friendly tool that is used for file format conversion, conformer generation, minimization of small molecules. All things can be done in OpenBabel. So you can go to this page called download. So Windows, of course, you have a GUI for that. And for Linux, you can directly install from this video, which I told you from here itself. You can directly install from the Synaptic Package Manager. OpenBabel can be installed. So you need not to do anything, just search for OpenBabel and install. Coming to Mac OS, of course, you have command line version, and the GUI version is called iBabel. So that's where the GUI version can be used. So this is a very user-friendly software, but a lot of flags available, optional flags, which is really, really helpful for merging multiple mol files to a single SDF file, splitting single SDF file to multiple mol files, converting PDB files to SDF or SDF to PDB. All sorts of file formats and minimization confirmation analysis could be done with OpenBabel. So considering I have all these installations have been already done in this machine, I'm currently using Linux. I'm currently using Linux Mint 64-bit on a machine with the 32 core and around 32 GB RAM. But of course, I'm going to run all these calculations only on one core, ensuring that you also run on one core. So before moving forward with the next steps, I wanted to refer to you tutorial which I have created in 2006 or I think 2008 or 9, workshop on molecular modeling AutoDock. So here you have a flow chart on where to start, what kind of files are required, how do you do analysis, and what are the each step. This particular tutorial is completely adapted from the original AutoDock tutorial. So it is available in scribd.com, scribd.com/GiriBio. You will get more details from there. So if you want, after this workshop training, if you want more things to be explored, you can refer to this particular web page. Anyway, so now we are going to start AutoDock. So let me just shoot it. So as I told you, I have already done some analysis, and I'm just opening AutoDock. This is what you see when you open AutoDock. So let me clear my screen so that for a better visibility. Okay. Okay. Things looks good. So this is how the AutoDock version. You might see a black window. It's just I made it a custom setting. Anyway, you can make it white color also. Now, there are several ways in order to bring in your protein into it. Let me tell the first manual way, which I usually prefer. Let me show you that. Where's D1DWD? So I have downloaded the PDB file. This is the PDB, original PDB file, which I downloaded from the internet. And now, if you wanted to manually create your protein file, it's easy from here to create. For whatever you see with the atom, just whatever starting with atom, you can see this is a PDB file. The line or the row which starts with the atom tells you about the chain where it is the protein is starting. So this is the amino acid 3-letter code, and this is the chain L. I already know which chain I have to work on. I have to work on chain H. Okay, so I don't want L. I will probably delete it, and then I will go to H. Ensure that that particular chain terminates at the line called TER. So we do not want all this thing. We need only from this atom till where the H term TER, the terminal ends. So that's what I am looking for. So let me scroll down a bit faster so that we don't waste time. Yeah, here it goes. So this is the TER of H. So I will just copy paste only this as my protein in order to load this to my AutoDock, and then to convert it as PDB-QT after applying hydrogens as well as charges. Now comes, I wanted to do a template docking or reference docking. I already have a ligand here in this PDB. You can see here HET atom. So water molecules will also start as heteroatom, that is HET atom, as well as a ligand. So I can copy paste this particular HET atom and ensure that you don't copy paste your water molecule. We don't want. But keep in mind, without water molecule, we need this connect also. The connect is required here. For the chemical compounds, we require connect, but not in PDB. So please remember, do not keep connect in the PDB, because this will give error when you're trying to do AutoDock Vina. So ensure you make things very clear. So the ligand can have the connect, but not the PDB. This is one way of doing it, a manual way. But there's another easy way to do it in AutoDock itself. So you can go to File, Import. I recommend you not to do it right now because you might miss what is the options available. You can just listen to it and then note it down. You can go to Fetch from Web. If your machine is connected to internet, you can give the PDB ID here directly and click OK. It will download the PDB and display within AutoDock. Now, you remember you have L chain, H chain, and I chain here. I want to clean my PDB now. How do I do that? So you have to go to Select and click on Select from String. And then here we need to know which chain I have to delete and which chain I have to retain. So here I'll say go to chain. I'll say I don't want I do not want L. And then I will say add, click add and dismiss. You can see there are yellow dots in the screen where it is being displaying which are the atoms and chains have been chosen. Then I'll go to edit and I'll say delete selected atoms. So these are the chains that got deleted now. Okay, now there is only H chain and water molecule. In order to remove water molecule, I go to edit and say delete water. So now water molecules are also deleted. You can see at the bottom there are the sequence single letter code is there. So there is no water molecule now, all are being removed. Now we have one more thing here. In the H chain, we have the ligand also. You can see here at the end. So if I want to make it display, let me make it as a display here. So you can see this is the ligand, co-crystal ligand that I have, that is MID in the structure. Now, how did I do all these things? Just to scroll your wheel mouse, then it will scroll up and scroll down and zoom in and zoom out, and use your left mouse button in order to transform or move around. Use your right button in order to turn around. Okay, these are the options available in your window available. Now, so here I do not want my ligand in my protein. I just need only my amino acids. So what I do is either I can define this as a ligand or I just remove it, both ways. So easiest way I will tell you in order not to confuse anything. Just again select from trim and ensure that you delete all the chain and go to atom set and select heteroatom. Keep in mind, you have to remove water molecules before doing this step because water molecule is also a heteroatom. And then add, now your ligand is also selected. You can see it is like a selected mesh. And then you can go to edit and say delete and then delete selected atoms. Now the ligand is gone. Now it's just protein, nothing else. So the same thing what you're doing in the text, same can be done in MGL Tools also. Now, before saving this as a PDBQT file, there are a few steps to be carried out. The hydrogen atoms are not visible during X-ray crystallography. So we have to add hydrogen atoms. There are several algorithms available. But in this case, I'll be going to add hydrogens by going to edit menu, hydrogens, add. Ensure that non-bond order, that is for PDB files, is checked. So whatever the default is there, just leave it and click OK. Now you can see the hydrogens are added. You can see there's a white terminal atoms that are added to most of the atoms. So that's where the hydrogens are added. Next is we have to also add charges. I'll tell you why we are doing this process. So go to charges, go to edit menu, then charges, and click on add Coleman charges. So here says Coleman charges added 6.67. And then we have to add one more charge, that is Gastiger's charges, that is compute Gastiger charges, and click OK. The purpose of doing this is when you convert your PDB file to PDBQT, now my question is, what is this Q and T? The Q means Quaternion and T is for torsions. So in order to understand AutoDock algorithm, the American algorithm, to understand what are the positions of atoms and interaction points, they wanted to understand, also calculate dielectrics, electrostatic field. So they wanted to understand what are the charges assigned for each atoms, as well as the torsions on different bonds. So we try to compute it, and when we, with this information, when we convert our PDB file to PDBQT, keep in mind there are two different columns are getting added. One is for charges and the other is for torsions. So that is what you mean by PDBQT. Now I am going to save this as PDBQT file, then save, write PDBQT. When you're writing as PDBQT in this particular window, please be careful. You can see here there is connect here. PDB records that to be saved. Connect is here. Ensure that this connect is being removed. Do not keep that connect. Because if you're doing AutoDock Vina, connect will give you error. So ensure that you click that and remove it. All the others are fine. And then you save it as a PDBQT file. You can see here. Now, one more thing I wanted to keep before moving forward, I want to tell you a few points. When you're doing any AutoDock calculations or AutoDock Vina or MGL tools, let it be Windows, Linux, Mac, ensure that none of the installation folder or calculation running folder has any space concatenation. So there shouldn't be any space should be there. Either it should be with an underscore or a continuous file name. So keep that in mind. In most of the cases, when people are telling me that there are errors while they are doing AutoDock or AutoGrid, it's only because there's a space in the file name or in the folder. So whatever folder or whatever file name on the previous, wherever you have installed, there shouldn't be space anywhere. So keep that in mind. So this I have already saved, so I'm not going to do it. This is the step. Now, one more step we have to do. Every time when you are trying to open the files into AutoCAD, AutoDock, you have to specify the path. In order to avoid that, what you can do is you can go to File and then Preferences and then click on Set. And here you have an option called Startup Directory. That means where should always when you click Open or Save, where should the software should look for which directory. So here we are looking for, I have put it in results, ADT, 1DWDD, MID. That is the folder that I've set. So you just copy paste that path, click on set, and then click on make default. So next time when you do a different exercise, you come up with the same thing again. So you have to redo this. Otherwise, every time the software will be going only to that directory. So keep that in mind. And then I said that. Now, coming to the left-hand side of this particular the tree diagram, if you want to delete something, you can click here and click the right button and say delete. If you want to release this particular menu, you click on dismiss. Now, here we are displaying in the manner of lines. If you do not want to display anything, you just click this. Next is if you want to display as a stick model, as you can see here in the screen, it is a stick model. If you want to display them as a CPK model, this is a CPK model. If you want to display them as a ribbon, the same thing can be done. If you want to display them as a surface, the same thing can be done. Now, let's say I'm putting a ribbon, and if you want to color it in a different way. So for that, what you have to do is you have to click this at the end, display coloring menu. You click this, and you can see how you want it to color. So whether you want to color as a residue type, or by the chain, or molecule, or polarity. So those things can be colored. And then you click on apply. You can see the color has changed now. So you can choose what kind of coloring methodology to be applied, or if you want a rainbow kind, you can apply and it's a rainbow kind. So this is how you can, so I know that many of you use PyMol, but many of the features can be also done here. That's what I wanted to show while going through these options. Anyway, so I'm going back to this step where you have the ribbon as well as the wireframe kind of representation. Now I'm trying to remove this. We have once we have prepared all the files, one with the DWD, and if you wanted to create a file for the Ligand file, again either your Ligand should be saved as a PDB file, PDB Qt file, or Mol2 file. So Mol2 files also being accepted as the Ligand file. Keep in mind, all your Ligand molecules should be in 3D geometry, not 2D. So before doing anything, you can use OpenBabel to convert from 2D to 3D by using the flag --gen 3D. And then you can convert a 2D mode or SDF or PDB, whatever it is to convert to mole to 3D or SDF. So keeping all this in mind, I'm just starting from fresh. So after creating all the files from the top menu, I'm going to the bottom menu where it is saying ADT 4.2, that is AutoDock Tools 4.2. I'm going to the Ligand, I'm going to input and click on open. So I have the Ligand here, which is already I have prepared as mid.pdbqt, which I have already prepared using the same method which I told you. And then I click on open. Now you can see the PDB charges are added, PDBQT charges are added. found the 16 aromatic carbons and detected 12 rotatable bonds. This is very important. The rotatable bonds refers us to understand the torsions as well as the possibility of generating conformations. And then torsions also defined to 10 and click OK. After that, you have to go to output and then save the PDB-QT. So, for example, if you're loading a PDB file, let me show you. If you go to Ligand, Input, Open, you can choose here to either choose mol2 file or PDB file, as I told you earlier. If you have a mol2 file also, that is also fine, or a PDB file, that is also fine. So you can bring them here, but finally, when you're saving this file, it is very important that you save it as PDB-QT. Make sure that you do that. That is very, because as I told you, it has to add the charges as well as the torsions. And then you can save them. Here it's already there, just overwriting it. Now the ligand is ready. As you can see, the ligand is being displayed here. If you want the ligand to be displayed in a different way, you can make that too, display it here. You can see that, right? Now, next we have to load our protein. In order to load our protein, we have to go to grid, macromolecule, and click on open. Here also, we have already prepared our PDB-QT protein from the earlier steps. Hope you remember what we have done a few minutes back. So from that, we choose a PDB-QT, click open. Now it will ask a question whether you wanted to preserve the charges that we have already done or not. I would go for no. You can also choose yes, but I would prefer to go for no, so that you will see the process, what it is trying to do. So it is saying that there are 5 residues which is having a non-integral charges for these many residues, but these charges will be corrected when the PDB-QT file is written. So automatically it will overwrite the PDB-QT and you can click OK. Let's imagine that, so there are no non-bonded atoms. It is very important that your protein should not have non-bonded atoms. If the non-bonded atoms are there, ensure that you neutralize them with the charges. So you can modify the charges. It is clearly explained in the tutorial which I referred to, how to modify the charges to make it neutral. And then click OK. Now, why this is happening? Usually, when you go from macromolecule open, there is an option that you can open PDB files. So when you open a PDB file, the software will automatically take care of adding the hydrogens as well as then what to say, trying to add torsions and then clean it and add possible charges to neutralize it for all non-bond bonded or for all bonded atoms, and then they ask you to save it as PDB-QT. But here, since I already converted it as a PDB-QT, it didn't ask for it to save it automatically. So there are possible ways, as you have seen already several videos. There are different methods that you can bring in. So I am trying to cover up all together. So I have loaded my macromolecule. The next is I wanted to choose my map type. How should I generate my grid map? And based on what? Whether I can choose directly or based on a ligand. Now, directly, I would recommend if you're working on a hypothetical protein where you don't know where is exactly the protein has the pocket, or there are several pockets which you are not having any idea from literature or any other solution, so that you are trying to make it, what are the possible ways you can make out of it, so that you can make directly. So I have seen some videos already. People try to make the grid box throughout the protein completely, so that wherever it tries to dock, it docks. That is something called as blind docking. Personally, I don't prefer that, because we have to know before doing anything what is actually we are trying to do. The reason behind that is, why can't you do it one by one rather than doing it all together? There's a big reason why I'm telling you this. The interaction points will be, the number of points will be huge when you try to define a blind docking and a larger grid map. When you have a larger number of interaction points, ensure that you also give larger number of genetic algorithm runs or iterations. If you don't give large number of iterations, then the possibilities of checking all the possible pockets in your protein is very minimal. So keep that in mind. Genetic algorithm, when you repeat the experiment, there are chances that you don't get the same result. That is the beauty of genetic algorithm. the iterations goes on generating and populating mutations, and it's a machine learning approach anyway, but they try to do it in order to make it much feasible to understand which are the possible interacting atoms. The second method is that choose ligand, because I already have a co-crystal ligand. So it is always better and clever to, if you found that your object is also on the same kind of amino acids that you wanted to target, whether if you inhibit those amino acids, whether it is going to down-regulate or up-regulate a specific pathway, if you already know about that, then we will go with that ligand. So we are choosing a ligand because I know, because my amino acids are similar to the focused amino acids, are key amino acids are something that is interacting already with a co-crystal ligand. So this is my co-crystal ligand. I select them. So select co-crystal ligand, and then I go to grid again, and then I say grid box. When you come to grid box, let me zoom it a little bit. So after saving this grid box, I will open the session for questions. Then we will move on. Okay. So here you can see the grid box is somewhere, but I wanted to see whether that grid box in my clear near to my ligand. No, the ligand is outside and outside of the grid. But I want something the amino acids which is very close to these ligands. So what will I do is I'll go to center and say center on ligand. I already defined my ligand, right? So the software understood what is ligand, and you can see at the bottom there is an yellow color axis coordinates. They are representing the center of the molecule. Now you have to turn around and see the red color, the blue color, and yellow color. The red color represents the x dimension, the green color represents the y dimension, and the blue color represents the z dimension. These are the number of points in z dimensions, and these are the center of the grid box. These parameters are very important when you're trying to generate your config file for AutoDock Vina. So keep that in mind. Now you have to see, now there's one more thing that you have to keep in your mind. If you really know the pocket size and everything, it's good. Otherwise, you think there are possibility the pocket might be slightly more bigger, because I have certain other ligands which is slightly more bigger. If you think that, you have to ensure that you increase your grid size accordingly. You cannot simply increase, but make sure that where you wanted to increase. Now, for example, in order to understand that, you can expand your protein and understand what are those amino acids. Okay, which amino acid I have to more emphasize and look at. So if I'm example, if I wanted to look at the 48 amino acids, I think it was serine. Yeah, serine. So if I click on this, I can see there is a surface that should have been generated. Let's see where it has been generated. I don't see. Sorry. Did I check it properly? Oh yeah. Let me take this off. Yeah, see, serine is here. You can see. This is what I was trying to just look at it. This is one of another active site of this protein actually, based on the pro site. There were many other sites also anyway. So like that, if you think that there are specific amino acids I'm looking forward, I can pick up those amino acids. So if you want to know that what are those specific amino acids, there's one more way. You can go back to the PDB and go to the bottom and click on this picture. This is a 2D depiction of the interaction between, sorry, where is that? Not this one. Okay, they removed it. Okay, I'm not sure whether we will be able to see it. There is a 2D. Let me check if it loads. Not this. Just a minute. It was earlier there. Oh yeah, I think this is the one. No? Okay, this is not being shown here. Usually it comes here, but I don't know what they have made the changes. So there it comes like a 2D depiction so that we will be able to know which are the interacting amino acids for those ligands. Anyway, I'll be anyway showing you how to get that. So by understanding that, if you really do not have any co-crystal ligand, but you want to just the receptors, or sorry, the residues you have to define, you can come up to on your left-hand side and then enable the surfaces for those residues, and then you will be able to define a grid there. If you think that this is your pocket, you can define a grid there. But that depends. Now we have a ligand, co-crystal ligand, so we may not do that exercise. But if you think that you have a hypothetical protein where you do not know what is a co-crystal and what is a site, you can do that exercise. Anyway, so here I'm continuing with the grid box definition. So here I say sender on ligand. Now, let me also do an analysis. Okay, let me create a surface for the ligand to understand whether it is still fitting within this grid. Now, you can see here at the side, there is a slight, your ligand is coming out of the grid. I hope you can see that, right? So the surface topology, actually, that particular ligand is coming out of the grid box, and this side it's also slightly coming out. You can see that there is a white projection that is coming up, but on the top, the blue areas, they are fine, and on the red areas also they are fine. They're completely fitting. So there are chances that you can make a bit larger on the Y. So I wanted to cover those which were being outside. I can move this. This can be used for moving it. So I just moved it and covered it. So, no, still there is a little bit. So I'm just covering that up. Okay. So now your ligand is completely within the box. So this level of analysis you have to do so that your grid will be defined properly. Now, you have to go to file and say output grid dimensions file. Very, very important in order to do AutoDock Vina. The center of the grid as well as the grid points can be defined only from this file. It can be also taken from this picture where whatever you're seeing here, but otherwise this is the best way. So I'm saving it as a grid.dim.txt. It can be any name, but ensure that you give the extension file also. After that, I will say file close saving current. Okay, now everything is saved. Your ligand is still there. Okay, now I go to I go to grid and then I go to output and say save GPF. GPF means grid parameter file. I'll show you what is a grid parameter file. It contains what all so far we have done, what all output has to be generated, what kind of atom types we have defined based on the ligand, or as well as on the receptor side, those things we are trying to define. So I just give a name as grid.gpf. GPF is for grid parameter file, and I saved it. So I have already saved it. Now let me show you that file. So here, this is the grid dim.txt, where it tries to tell you what is the space, what is the points, the x, y, z points, and as well as the sender. So this same data you can see in the grid.gpf file. So this file is called GPF, that is grid parameter file, where just a minute. Okay, I would recommend everybody to please switch off your webcam because some of the participants are not getting their bandwidth. So it would be great if you can switch off your webcam. Okay. Okay, great. So, okay. So here you can see these are the points, the grid points that we defined. Here we are trying to save the field map. This is the spacing. These are the different receptor types. There are hydrogen bond donor, hydrophobic, then acceptors, and many more. And then these are the different ligand types available. The receptor file name is defined. This is the grid sender, which we have it in grid dim. This is the grid sender. So grid sender is also defined here.
how we want to store a minimum energy within the specific radii. So this is zero point 0.5 Armstrong. And then for each atom type, we are generating the map. This is only required for AutoDock. We don't require this for AutoDock Vina. And then we have dielectric constant, desolvation maps, and electrostatic maps. All these maps are required for the AutoDock to generate the free binding energy. The free binding energy is a combination of van der Waals, electrostatic, and many more other energies. Anyway, so we have reached to the level of grid parameter file. So after saving the grid parameter file on the same folder, we have to go to run, click on AutoGrid, run AutoGrid. Keep in mind, in order to work this AutoGrid in the same way as it you see here, you have to initialize or set the environment variable for AutoGrid, AutoDock, and AutoDock Vina throughout your machine, throughout your operating system. For that, for Linux, I can show you something very easy. So if you see, these are the commands. Now, home, GDBio, softs, MGL tools, that can change in your machine because you might be putting in different folder or different user, we don't know. But rest of the things are same. Now this is available in the README file of the installation folder. So you need not to search this anywhere. When you install the software after installation, there's something called README. In the README file, it is clearly mentioned, please put this for a Linux machine. And for Windows machine also, they are clearly mentioned. Go to your system settings, go to set environment variables, and you can put all these there. We don't require all. You need only the main thing is the first line, then this ADT, and the last line. Sorry, last two lines. That's all you require. You don't require the other too much because I never use PMV and the visions. So, so if you set this, then it's easy for you to put this AutoGrid for here. It works fine. But if you do not have set, you feel that, oh, it's very difficult to do it, I don't want to do it. The best way to do is AutoDock Grid file and AutoDock file. There are two executable files when you're downloading it. You just put them in this folder, the folder which we have set already, where these PDB-QT, PDB files all are being saved in a folder. No? You just put it there. After putting it there, you click on browse and then just click the AutoGrid file there, so that the full path will come anyway. So for example, if it goes to that particular folder and then I click a single file, let's say it was AutoGrid. I don't have it here. AutoGrid 4. You just click it and say open, so that it will come here with the path /home shastra softs result ADT 1DWD underscore mid /AutoGrid 4. So these are the two ways you can use AutoGrid. Ensure there is no space, there is no gap in file names as well as in folders. You have already seen this, my path, there is no space. Now comes a parameter file. Sometimes already the file might be loaded there, but still you have to redo it because your log file should be generated. So we have to take the GPF file. You browse and your grid.gpf file to be opened. Now you can see there's a GLG file is also generated. GLG means grid log, the log file that contains all the data, output data. And then you can click on launch and the grid AutoGrid is now running. It completed anyway. But before moving forward, if you have any questions, please let me know so that we will clear that and I will proceed with the next. You can unmute yourself and ask questions. Any questions? No questions? Can I move forward then?
Hello.
Hello? Yeah.
Hello, sir. This is Praveen.
Yeah, Praveen. Tell me.
Sir, I want to know whether the charges which we apply for ligand as well as for protein, both should be the same charge, or on what basis we should?
See, the adding, the proteins as well as for the ligand, for the bonded atoms, the charges are being computed using gas-to-gas charges. That is no problem. We need not to take care of it. If there is any non-bonded atoms, for example, you have sodium, or if you have any zinc atom in your protein, There they might be having 2 plus or some 2 minus or 1 minus charges will be there. That you have to manually correct it so that you make it neutral. That though I don't say it's a drawback, but we have to intervene to make the system neutral. So you are not giving any charges to ligand manually or to protein manually. That is computed through Gastiger's charges. If there is only non-bonded atoms in your protein, that's where we have to check the charges to make it neutral.
Thank you.
Okay.
Thank you, sir.
Hello.
Hello.
Is that okay, Praveen?
Yes, sir. Okay. Thank you.
Any other questions from others? Okay, that's good. So I think I covered most of your questions then so far. Anyway, so I will proceed with the next steps then. So before moving to the next step, we wanted to see what all the files have been generated. So as I told you, we already have a grid.gpf, that is a grid parameter file. Now from that we generated grid.glg, that is a log file. Let me open that file. So this is a grid log file. So when we open, you can see there are a lot of torsion analysis has been conducted, and they try to define the type and all, and also calculated the energies between different interactions, and they were trying to see what are the different atom types on a pairwise interaction energy within zero 0.5 Armstrong, and they calculated the hydrogen donor, hydrogen acceptor energies, and based on all these calculations, they finally came up with 8 grid maps for electrostatic potential, desolvation potentials, as well as hydrogen donor, and many more other atom types. So negatively charged, positively charged, donors, acceptors, all those being carried forward, and it should show that AutoGrid for successful completed. If there is any error, it should pop up, but if it is completed, you should see like this. By the way, I'm extremely sorry, I forgot to mention one very important thing. Before, when we were generating this particular file to save this protein as a, PDBQT, you have to do one more step, because I already mentioned that you have to add hydrogens. You also have to check the charges. There is one more step left. That is, you have to go to edit atoms and say assign AD4 type, that is AutoDock 4 type. You have to do this before saving this as PDBQT. It's very, very important. Since I have already saved it and kept it ready, that's why it didn't warn me. Otherwise, it should have warned. So keep in mind, after doing your cleaning process and all, deleting water and all this process, you will be doing adding hydrogens. Then you will be adding charges, that is Coleman charges, as well as compute Gastiger charges. And then you have to go to edit atoms, assign AD4, and then only you have to save your protein as PDBQT. Okay. Sorry, I just I missed it. Extremely sorry. Now, based on after doing the grid map, you can see these many files have been generated now. All these map and FLD files have been generated. All these files are required in order to understand how many interaction points, what are the possible, what to say, electrostatic fields available, and desolvation terms. All the factors are being embedded in this. They're all text files, nothing like binary, so it's readable also. So in order to understand what are these different file formats and all, it is there in the web already. Let me show you that. If you go to this particular website from Yale University, so you can see, let me go to the page, AutoDock file formats. I'll be putting all these links in the YouTube video description, so you will be able to easily access it. AutoDock file formats, what that each file format should contain, what is the difference. So they have clearly mentioned whatever I'm telling you, the same and much more is being there. GPF file, and what are the different keywords that is used in GPF file, the grid, what is there in the grid map file, and grid map field files. So each one is very well explained here. Let me tell you, we will be urging to finish off our docking calculation, but unless you understand these parameters, you will end up, you won't be able to write your description of your discussion part of your paper or your report very nicely, because change of a grid sender drastically changes your interactions also. So keep that in mind. So this is one reference that I wanted to give you with related to files and formats, which I was showing you in the agenda of this meeting. Okay, going back, after generating grid, everything is fine. Your file is successfully completed. We checked everything. Now we are going to the next part, to docking. In order to go for docking, you have to first choose your or load your macromolecule again. So I just say macromolecule set rigid file name. I know this is my which I loaded, so I just clicked it, so that ensure that there is only two parameters here. One is my ligand, the other is my protein, and this ligand is used only to generate my grid file. When I'm doing AutoDock Vina, I can bring in any other molecules, no problem. And then I go to docking again. I go to ligand. I say choose ligand, and I choose MID value, MID that is already being loaded here, and select ligand. Now it is asking me something. Here also you have to be very careful. Now, most of the time we being learned that it is going for default values. So here there are different atom types, and center of the ligand molecule we have a different value here. You remember we have the center of the grid box slightly different because we modified it. If you have modified it, ensure that you uncheck this. That means the user specified initial position, it will start from here because we modified it. If you did not modify it, it is fine. But if you modified it, I modified it because I saw that the grid was not completely fitting in with my ligand. So I just covered it with all the surface accessible area. And then I uncheck the random points, and therefore it starts from this point. This is very, very important when you're trying to do a template docking. Template docking in the sense you wanted to redock your existing co-crystal ligand and try to see whether your parameterization in the AutoDock for this particular protein is good or bad. Of course, this is a validation kind of a step or a redocking step. And then I accept it. Okay. And then I will go to search parameters. I have to choose genetic algorithm. I am not covering any other algorithms as of today. So genetic algorithm. Here is what I was telling you. Most of the time we go for 10 genetic runs. I would recommend 10, more than 1,000, 1,000, or if you're going for publication, at least 10,000. I agree your machine will take at least 7 hours or 6 hours to complete the job. But if you're submitting in a very good peer-reviewed journals, let it be any, I'm not ranking any journals based on impact factor, but a good reviewer, he or she will ask for minimum of 1,000 runs. The reason I told you earlier, if you have your pocket is larger, and if you say only 10 confirmations, the possibilities of the docked confirmation is only 10. So it might not have covered the whole pocket which you have defined the grid size. So it is always recommended that you go for a more number. It takes time. Of course, analysis will take time. But we do not want to leave any possibilities. This is what we call it as missed opportunities. When you think that, oh, it takes too much time, I don't have time to do this, I will give only 10, then you're missing opportunities because the opportunity in the sense you're missing the right answer. That's what I'm trying to say. The maximum number of generations also to be maximum. Here also we try to reduce it most of the time. Please don't reduce that. So I would recommend to keep this 1,000 or 10,000 max. My favorite is 10,000. It takes time, but you do 10,000 only for the confirmatory ones. For example, after doing virtual screening, you know the best 3 are there. For 3, you do it. That will have a very significant difference compared to what you do for 10. When students who do your internship, they try to do for 10 and they say we didn't get the expected result. I asked them, okay, do it for 100 or 1,000, and then they come back and say, okay, yeah, now it is something favorable, makes sense. That is the difference with genetic algorithm. So I'm not going with each of this peculiar thing, but as I told you, this number refers to the docked confirmation, and this number, which is again maximum number of evaluation and also the maximum number of generation, also replicates on the interaction points. And then I accept it. and then I go to run, sorry, docking, and then I already did search parameter and docking parameters also I have doing now. So most of the time we are this docking parameter, we choose it as default. But let's go through what is docking parameter. Random number generation, whether you want it to be a platform independent library, of course, it will be from a different library or built-in library. Most of the time we go for a built-in the platform independent library, or you can user define it. Then energy parameters, what is the external grid energy that you want to give, or the available initial energy, and the maximum number of retries you wanted to give, whether you wanted to calculate internal electrostatic energy or not. So by default it is no. Then step size parameters. Step size parameters are something like we are referring to quatrion and torsions, how far step. So this is basically we are trying to see what kind of conformations, flexibility that is being given to the ligand. That is called step size parameters. We keep it as again default. And then coming to output format, whether you want a detailed output or what is the RMS cluster tolerance you want to give. It is a 2 Armstrong is a cluster tolerance. And then reference structure for RMS calculation, that is my co-crystal ligand, that is mid.pdbqt. And whether you want to perform a cluster analysis, by default, it will perform cluster analysis. Then whether you want to write all the confirmations or whether you do not want to write all confirmations, that can be also done. By default, it performs cluster analysis and it does not write all the confirmations. It writes only the best ones. So these are the ones which again I'm going for use default because to make it much faster. But I would recommend to please understand these parameters. They are really, really helpful when you're going for compounds with more rotatable bonds, more flexible compounds, or when you're working with the natural molecules with the large cycles, I mean cyclic forms, these will be very helpful generating the conformations. I just accept it. And then finally, I do not want to take this AutoDock 4.2 parameters. It is already inbuilt, so don't define that. And then go to output and say Lamerican genetic algorithm. So I'll be saving my DPF file, that is docking parameter file. So I have given as doc.dpf file. I save it. I have already saved it, so I'm not redoing it. Now let's see what is this DPF file. So if you go here, this is the DPF file. Here, AutoDock parameter file is already being taken. Then calculate internal electrostatics I have given, and then atom. Can someone of you help me? Where did you miss or up to which part you have heard? Could you please help me with that? Hello?
Preparing for running auto docs.
Oh, up to that. Okay. So, okay. Thank you. Okay, docking parameter file. Okay, thank you. Sorry. Technical issues. Anyway, so I was telling you about the AutoDock parameter file, and cluster analysis also will be considered. The same file can be used in uh, sorry, yeah. So the same parameter file can be used in command line. As I told you, referring to the tutorial, the same tutorial will include both the GUI version as well as the command line version. So for each respective step, the command line versions are also given. Going back to here, we will now do the docking part. So run dock. And the same thing which I already told you last time, that you have to either set the sources or environment variables. Otherwise, you can load AutoDock 4 and AutoGrid for files in this folder, and you can browse and take them. Otherwise, you can use it directly. And then a parameter file, I have to take the DPF file. So I have the DPF file and DLG file. DPF is the docking parameter file and DLG is the docking log file. And then I launch them and run them. So let me launch. I have already calculated, but I'm redoing it. So I launch them and it is running now. So the calculations is running. I just did it only for 10. Okay. So let it run. I will also show you for the config file for the AutoDock Vina. Then we will go to the result analysis. So,
Can we ask questions now?
Yeah, please go ahead. You can.
Yes, sir. Today I did a trial of 15 trials, 15 genetic algorithm runs.
Okay.
Autodoc.
Okay.
The results I got is like the better results within 10.
Okay.
And previously for the same protein and same ligand for the same grid file,
Yeah.
I did 10 trials. The maximum energy, like minimum energy, highest minimum energy was same in both the runs. I didn't see any much difference in running more number of
Okay.
runs.
Okay.
Now, okay, explain, sir, explain.
Okay. So when you say about the energy, can you tell me whether on both the runs they both interacted with the same amino acids? One, second, whether which one did interact with the key amino acids as per the literature or some other analysis?
Yeah, I can't say that, but
Yeah, this is because you have to do the analysis, which should be my amino acid before, because tomorrow when we want to discuss about docking, we need to discuss on biochemical terms. Okay, this atom has been going with this particular atom of this amino acid. If you inhibit this amino acid, this pathway could be down regulated. That's how the connection goes. Now what you say, I really agree with you, and I have already mentioned in my session that when you redo your docking with the same parameter, you will never get the same result. It might go bad or it might come better, because this is because of genetic algorithm. So more the number of runs, it's always the better, because you have all the possibilities.
okay, sir.
Yeah. Any other?
Yeah, but the end of the docking results is the person will ask what's the energy means. That's the final
I
result.
understand you, Gautam. May I know from which department you are?
Yeah, I'm from biotechnology.
Biotechnology. Understand.
Yes, sir.
See, I completely agree with your question, and I am very sorry for the situation. You're very right. Whomever we are talking to, they are asking only for the score or only for the energy.
Yes, sir.
But for me, energy and score is also important when other parameters are being looked at first. For me, the key amino acid is the first part. The second is whether there is any intra-ligand or inter-ligand clashes happening or not, whether my torsions are proper or not. Then only I will think about my energy and score. Because for energy and score, if I say minus 9.8, that is for that particular compound within that set. If I would have chosen a different ligand, I could have got minus 15 also. Right? I don't know. Within that data set, I didn't search for everything. I did not screen for million molecules. I screened might be for 15 or one molecule. So my parameter of selection will be more prior to understanding the mechanistic way, whether it is interacting with the key amino acid, whether all the other parameters are proper. Then only I'll think about the energy. Energy is an important factor, but when all others are being meaningful, then only energy makes sense.
Okay, sir.
Okay. Any other questions? Our docking is done, so we will go with the next part. But if you have any other, we can answer those questions. So shall I proceed?
Okay. Now, going to the next, if you wanted to do AutoDock Vina studies, keeping all this same, I'm going to docking and then I go to output and say AutoDock Vina config file. That is called config.txt. This can be manually also created. The advantage with this option is automatically it tells you what is the receptor, what is the ligand, what is your sender XYZ coordinates, and your size of the coordinates. Now here the size is a bit different. We will change it. Then what is your output file? And if you want to define a log file, the log file also can be defined. If your plan and the config file file name can be also defined. If you wanted to run in multiple CPUs, you can define how many number of CPUs or cores you wanted to mention. And you click on save, it will save the file. I already have saved this file. Let me go to that file. So here you will see config.txt. The receptor is here, the ligand is here, the sender X, Y, Z is here, and my X, Y, Z, the points, grid points, my out file, then my log file, that is Vina underscore log.txt, and exhaustiveness to be 8. That is by default. It's always 8 is recommended, or 10 also you can put. Now, where did these numbers come from? Let us go look at the GPF. Did I close the GPF file? Oh, it's here, GPF. So here you see 40, 46, 40. From this, you get this number. So I will go here. So I have 40, 40, I'll put 46 here. Okay. And then I have the points here, the grid center points here, 31, 14, 24. I think they are same here, 31, 14, 24. So these values are related anyway, and I can save this and then I can run. In order to run that, again, you can go to MGL Tools, AutoDock Tools, go to run and then say run AutoDock Vina. After doing AutoDock Vina, as I told you, either you should have AutoDock Vina in that same folder, the executable file, and then you have to also choose your config file. This is the one, the config file. That's it. It runs. After it running, I'll show you what all it is much faster than AutoDock actually. So let me show you the output. So this is what you actually get. This is all you see in AutoDock log.txt. So it tried to do 9 confirmations, and this is the binding affinity that is called kcal per mole. The distance from the first, this is what I defined as an initial geometry, and from there how far it is getting deviated, that is the RMSD value here, the best mode RMSD values also. Now, in order to visualize this data, we will be doing the next. So your log file contains this particular table, which tells you what is the free binding energy on the affinity terms in kcal per mole for the same molecule. Now, going back to here, let me first analyze AutoDock Vina results, and then we will go to AutoDock results. Do you want a break or something? I can wait for 2 minutes. It's already 6:30. It will go for another 20 minutes, or if you have more questions, 30 minutes. So let me know.
I think you can go on.
Okay, I'll go forward.
Thank you.
So going to analyze, keep in mind, in analyze, there are two things that to be considered. One is, imagine that you don't have protein or ligand in your loaded into the ADT, or even if you already have it. So first, what you have to load is go to analyze dockings and open, and you have to open the DLG file. Okay, this is the docking log file, and you open it, and you have to give a new name. Of course, if your co-crystal ligand is already there, it will automatically name it as -2, and then OK. Now it is asking, we have already read 10 docked confirmations. If you want to play this, you choose this option. So if I have given 1000, then 1000. So like that, it will be giving up. So I just click OK. Now, before going to do that step, I'll go to analyze, I'll go to macromolecule, and I will choose, don't open it. If your protein is already loaded into AutoDock Tools, do not open it because it will create another one more entry. If it is not loaded, you can do open. Otherwise, you just click on choose. You click choose and say this is your protein and select macro molecule. That's it. In some cases, when you load the DLG file, or when the DLG file also tells you what is the receptor, when you click on open, automatically it opens the file if the receptor is in the same folder. So ensure that all your results, data, everything is in the same folder. Automatically the software will take care of it. Now, after that, I am going to confirmations under analysis, play ranked by energy, that is free binding energy. So I'll keep this side. I will click this button in order to display more option, which opens the panel. So I'll keep this here. And then I click on show info. I'll keep this here. I hope everybody knows about these options, which tells you the binding energy, ligand efficiency, inhibition constant. I really do not recommend to report this inhibition constant. No, never. And inhibition constant units are in nanomolar. How can you be very definite with that number? When you're trying to do bioassay itself, we have to reiterate it to understand the standard error. So here without error, it's giving, so we cannot take that. So I don't recommend you to report these. But all other energy terms like intermolecular energy, van der Waals energy, electrostatic energy, total internal energy, torsional energy, unbound energy, they all make sense. Okay, keeping this aside, I'll go again to analyze, docking, and say show interactions. Another window will pop up and it cleans up, the script will clean up my window to show just the interacting residues and all. If you go here, there is something called display MSMS. I'll just unclick it so that the surface goes off. And then on atoms in close contact, I'll uncheck this so that wireframes goes off. Now this is bit very clear to me on which is my ligand and which is the residues. Okay, there are two things you can see. Let me color it properly. I wanted to understand what is my reference ligand. So let me color this in a different color so that you will get a reference point just for a representation purpose. So I'm saying my stick should be colored by molecule. So now you can see which is the co-crystal reference conformation and my docked conformation. The pink color or whatever you call magenta or cyan, that is the co-crystal ligand. And the other which you see neighboring is my docked ligand. This tells me that 10 was not enough. The 10 confirmations were not enough. I should at least run 100 because it should be much closer when I try to do redocking. And you will be able to see that there are interactions happening between the ligand and the amino acid, right? So that you are able to see. And the interactions are shown only for the docked ligand. If you think that the reference is really disturbing you, just switch it off. You can switch it off here as you can see in the left-hand side. You can switch it off, okay? So now you will see only your ligand here. Now, if you wanted to see what are the amino acids which are taking part, I have a glycine 216 which is interacting with the nitrogen of the ligand. That is what is happening. And I have other interactions taking place like hydrophobic interactions are also taking place. Now, if you think that I wanted to analyze and go through one by one, of course you can do that. So I'll click on this particular thing used to show frames via random access. I click next, all gets updated. What are the amino acids that gets updated? At the bottom, what are your energy values that also gets updated? So if I just click next, it goes on changing. You can see that the residues also gets updated and the confirmation also gets updated. But if you think that I wanted to see, I just want to sit back and relax and see what all the changes happening. And keeping my reference ligand also there. So I just click that button, it does it automatically for all the confirmations. So this is good if you are trying to do it for all. So immediately you can stop if the conformations are closer to the co-crystal ligand. Now the question comes, can you show the list of the conformers? Of course, you can click this, show the list, and that this will come, and you choose here, that will also, you have to double click, okay? When you double click, that will be displayed here. That is also possible. So these are the different the options available in AutoDock tools for you to result analysis. Now let's say you wanted to write or export this particular conformation. Let's say the first conformation, which is having an energy of minus 10.39 kcal per mole. Of course, you get the information here. So here, if I say write current, I can save it as a PDB-QT. Do not try to save it as PDB. Still, the file information will have PDB-QT. So when you're converting using OpenBabel from PDB-QT to SDF or PDB, that you have to define the input file as the type as PDB-QT itself. But in this moment, I wanted to refer to one more thing. In the AutoDock website, you have something called, is there a way to save protein ligand complex as a PDB file in AutoDock? Just execute these commands wherever the folder is there. The DLG, from the DLG file, it will generate all the PDB-QT file for the extracting the docking from your DLG file. If you want to convert a PDB-QT to PDB file, just execute this command. So so what it tries to do, it tries to remove the charges and torsions so that it converts to a PDB file. From PDB, you can convert it to SDF, mol2, whatever file you want. And if you want to split multiple PDB file into separate PDB files, for example, you have a protein as well as ligand, and if you want to split them up, this is the script that you have to use. This is a very easy script and nothing specific to any software. If you want to create a PDB complex, you just say your receptor and your result docking and you merge them. That is called the complex PDB. It's a text file, no? We are just combining it. When I showed you, when we started the session, I showed you manually you can compile this. The same thing we are trying to do using scripts. So nothing to worry about these scripts, and also these will be very much convenient. I hope you are able to hear me. If there is any issue with the audio, please let me know. And just a minute, let me just switch on. Okay, great. So this is all for the file conversions which I wanted to refer. Now let me go back to AutoDock. Okay, so I will close everything now because this is with respect to the AutoDock. Oh yeah, one more thing. To analyze with the clustering. So you can go to clustering and say show. So you will see the clustering that has happened. Here you have two confirmations. Here you have three confirmations. Based on the two confirmations, you have energy of in the range of minus 10 point something, which we know that here you see the binding energy range in this cluster of two is 1.13. So the binding energy is minus 10.39. So if I click this cluster, which has three of them, so it loads saying that the cluster of three is 0.78 and the energy is minus 9.06. So this is the advantage of genetic algorithm. It tries to cluster and come up with a confined conformer with the data. Okay, so I just closing all this data because I wanted to show you AutoDock Vina results also. And I just delete this, right? And I also delete this because I will reload them now. Okay, so now I am going to load my ligand as well, the protein. So I just say open, let me open the docking result first. That is from AutoDock Vina, okay? So I'm going to docking, open AutoDock Vina result, and then I say, this is my AutoDock Vina result, mid out. I just open it then it shows you multiple molecules or single molecule with multiple conformation. In this case, it is only single molecule with multiple conformation. If you have multiple molecules, for example, if you did the Pyrex or script-based virtual screening, then you have to select multiple molecules. So you just click this one for this case, and I click OK. So there are 9 docked conformations being loaded. That is completely fine. And you can see the kcal per mole, the binding energy is also being displayed here as minus 9.7. And now there's no buttons on the screen. You just use your keyboard left and right to go through the conformation changes. You can see the conformations are changing, right? Now, the problem here is the software will not show you interacting partners or anything because it does not have information like what AutoDock has. But I still can load, I don't know whether I can load it again from here. Let me try it here. Yeah, I can load the protein. So even after loading the protein, let me, make it as a surface. Great. Yeah, now I am here. So you can see this. This is the AutoDock Vina results. I'll explain it. Whatever you see in the pink color or magenta cyan color, they are the co-crystal conformer. And whatever you see in the green color, that is the AutoDock Vina results. And this is the pocket, okay? You can see it's very nicely visible. And now if I go through the keyboard, like left and right keys, you can see the next conformer with the minus 8.9 kcal per mole. The next one, minus 8.6, 8.5, 8.2, like that it proceeds. Now, how can you export this file? It is very much close, but not really great. I have to do it a little bit more extensively so that I can get much more closer confirmation. How can you convert this PDBQT to SDF? Of course, you can use OpenBabel. So using OpenBabel, you can convert it to SDF. I would like to display the SDF file that is which was converted earlier. So let me go to the command line to open a free software called as Data Warrior. So let me try to open that. CD soft data, warrior, data warrior, right? Oh, I need not to. I can directly open it. Just a minute. Let me go to that folder first. That would be better. Results, edit. Yeah. From here, I'll try to take it. And warrior, data warrior. So this is just this software can be used to display SDF files and all, and some comparative analysis. Not much of analysis could be done, but yeah, it helps. It is really helpful for your data analysis. Now I'll open the recent one, which I have converted to SDF, that is vina.sdf. You can see here in the remarks, Vina result minus 9.7. Hope you remember we have minus 9.7 here. So this is displayed here. The second one is minus 8.9. You can see here minus 8.9. So I just converted the PDB Qt file, that is which is output of Vina file, to a SDF file. So these files, of course, you can export it and you can modify it. Now, the next part of my analysis is that how can I generate 2D depiction of this particular results, which is really important because 3D structure alone cannot be feasible. But before that, I wanted to show you that for this particular surface, how can we make this surface with the polarity? So it's very important that when you're defining a surface of the pocket, you need to define the polarity or hydrophobicity or lipophilicity. But in this, I don't see any hydrophobicity other than polarity. So you can color it according to polarity to understand the negatively and positively charged regions, to talk about lipophilicity and hydrophobicity, off the pocket to discuss more about the interactions of the ligand. So going back to 2D depiction, I have my choice is two different websites. It's freely available. One is called PostView from Protein Plus. It's online available. You have to just load your PDB and you have to load the SDF file of your ligand. I have already loaded it in order not to have any delay in the internet. I just select this as my ligand, which is already docked. As you can see here, the log docked ligand. You can load your original PDB itself, so no problem. As you can see, there are two other chains also available here. IL chains are also available here. So you're fine with loading your original PDB because our main concern is the conformer of your ligand should go well. And after that, you should click on the post view, ensure that you have Java enabled browser, and then it should show you a 2D depiction of the interaction between the ligand and the protein. I highly recommend when you publish any papers, you have to include this 2D depiction in your manuscript rather than the 3D pictures. Of course, 3D pictures gives you more explanation about hydrophobicity and lipophilicity, position, chemical exploration, space exploration, how far the RMSD values are better, all those things can be explained. But to understand the interaction points, this gives you much more understanding of feasibility. Sorry, I don't know why it is taking too much time. Usually it doesn't take much time. Maybe there might be a Java issue. Let me check. I think everything is fine. Sorry, I don't know what happened. Uh, Java issue probably. How it looks like, I will show you. I want to show you an example how it looks like. Uh, Post view, yes. It is free to academics anyway. So this is how it is going to look like. So you will get a picture like this, which you usually see in PDB. So this will be a ligand and you will see the amino acids surrounding it with the hydrogen bonds, and the green cap will represent the hydrophobic regions. So this gives a beautiful 2D depiction of your interaction. You use any software for docking, but you can use this interface to generate the 2D depiction. Then my second choice is a ligand ligplot. So ligplot also does the same way, but not so clean. A lot of drawings and here and there. So it's your personal choice which one you want to go for, this or that. Both does a similar job, but this looks more clean and more intact. So keeping all in this mind, these are the kinds of analysis. Um, so um, some of you were asking for manual. As I said, this is the website. You have to go to script.com/giribio. Whatever today I discussed, all the details with the command line access, everything is there in this tutorial. There are plenty of other tutorials. They are also good. I don't say no, but it's your choice. You can go multiple tutorials helps you. So today, whatever I showed you, the demo, actually, I have referred to multiple tutorials and came to a conclusion. That's why I showed you how to create your PDB Qt file in several ways, either through command line or through just editing the text or through AutoDock tools and modifying and removing the different chains, and you need to know which is the chain you actually wanted to, because if you're working on a homo like a dimer, let's say there are two chains, you need to know which chain to be retained. If your ligand is between the two chains, you need to know that the two chains to be retained. So a better understanding about which chain to be retained, which amino acids you have to interact, these are all very trivial. So keep that in mind. And before doing any docking analysis, you should have a better understanding of what is your target. So that is very trivial. And if you have any questions, I'm happy to take it over. So now it's a discussion time. You can unmute yourself and then go ahead.
Hello, Girina sir. This is Dr. Kamli from Nanded.
Hi, sir. Please.
Yeah.
Oh, sorry.
My question is regarding the energies
Yeah.
docking score. Is it really necessary every time that we should refer to a standard?
Not necessary, sir. Let me show you an example for that. Because in some cases you won't be having references. So let me show you that. So just a minute. There's some. So let me go to that slide. This is slideshare.net/giribio. So I have a slide just to tell you about the docking parameters. This also tells some more detail about OpenBabel. Yes, this is the one. I hope you are able to see it. So when you're talking about molecular docking, as Sir was asking about energy, do we need any? Okay, just a minute. Are we all connected? Yes. Do we need any reference to any energy? Really not. What we are trying to see, the energy helps you to understand how far your specific interaction from an atom is contributing to that total energy, whether it is negatively contributing or positively contributing. That is what is very important. So we cannot say that, okay, if the energy is this level, this is going to be very good. No. For example, in your data set, let's say you have 15 compounds. Among these 15 compounds, which one is having the highest negative value? That is what we are going to look at. There is no standard threshold that is being defined. A standard threshold can be defined for a specific target if you have done some analysis or if you have a reference molecule. If you don't have a reference molecule, apart from energy, these are the other parameters that you have to consider. Catalytic function, regulatory mechanism, cleft detection, the right cleft detection, geometric analysis, the flexibility, conservation, characteristics, and then we think about the interaction energy. So if you don't have a reference, then it's always better to have a, what to say, a collective conclusion on which is going to be better or less. Now let's say energy is the ultimate term that we have to consider for choosing a compound or not. The answer is up to 80% yes. 20% is looking at the interaction points also. So even if you don't have a reference, then we have to see among the set that you have considered, which one is giving optimal highest negative energy with the interactions with the key amino acids, as well as there is no clashes among, that is intra-ligand, intra-protein, or inter-ligand interactions, clashes. This shouldn't be there. If they are there, then your energy is wrong. So that you have to make sure. I hope I answered your question.
Yes, sir. Thank you.
Thank you. Any other questions?
Yes, sir.
Yeah.
Sir, let's consider like recent outbreak, pandemic, COVID-19.
Yeah.
If I want to search or work on COVID-19 surface proteins,
Yeah.
like suggestions I got, like that's a new virus. No, lot of research on that done on that.
Yeah.
So you might not be able to get accurate result if you consider for academics.
Okay.
So it's a considering factor to take a new virus or new protein that has been to deposited in PDB.
Okay.
Yeah, that's it, sir.
Okay. So for that answer, when you started your question itself, I went to this website. So this is a GitHub, openly accessible to everybody. I have created a repository called COVID-19. You go to that repository, and then you go to proteins. This is an open project, never published anywhere. If you want to cite it, you can cite it. So you go to proteins. If you go to proteins, and if you go to the
text mode, I have clearly mentioned what is the regions of COVID, SARS-CoV-2. COVID-19 is a disease name anyway. So SARS-CoV-2, this is the ORF1A, ORF1B, then structural protein. So membrane, outside, and in interior regions. And I have clearly mentioned what are the reference PDB IDs. For 3CL protease, this is a PDB ID. Non-structural proteins, of course, we don't have a PDB ID, but these are the inhibitors that we have already people are working on. RNA-dependent RNA polymerase 6M71 is a PDB ID. Helicase, of course, we don't have anything. And endoribonuclease, of course, we have this PDB ID. So S protein bound to AC2, we have this PDB ID. Spike protein, we have this PDB ID. So there are plenty of PDB IDs available. These are taken from a Nature paper. So, and also at the bottom, I have already referred what is the relationship between one PDB with the other PDB. If you go up, there's a text file which tells you, for example, 6Y84 or 6YB7. If I go to 6YB7, it tells you how far 6YB7 is related to 6Y84. These are the PDB IDs. So I am having an RMSD of 0.1 with an Z score of 49.6. And there is a number of residues are also different from 6YB7 and 6Y84. So already we did some analysis on different PDBs. So if you wanted to know which are the right PDBs in order to carry out any structure-based analysis, of course, you can go to this site. It's openly accessible. So you have to go to www.github.com/vdbio, and then you will see COVID-19 in that proteins folder. Hope I answered your question now.
Yes, sir.
Okay, any other? In the chat, what can be done when the active site is not known instead of blind docking? Anusha, it's a good question, but as I told you, when you try to use Caspi server, they try to find out what are the possible unoccupied pockets. They don't look into any residues, but they just look at unoccupied spaces or pockets. So you try to do that first, or using Pocket Finder, online free tools available. Or even if you go to this website, there is something called as a doc site server. Let me show you. The one which I just closed. Where's that? Yeah, this one. So if you go here, let me take this PDB, 1DWD. Sorry about that. Something happened. Oh yeah. So one DWD, and I just go. If you go here, there's something called doc site scorer, binding site detection. You can use this tool. It is also free. So you can use this tool to go for a protein where you do not know what is your binding site. And then they might come up with the 2 or 3 binding pockets. Try to dock on all those pockets rather than doing blind docking, because blind docking is waste of time, waste of resources, unless you have there's a purpose for that. When you are having tools to find these pockets available, let us focus on those pockets and understand there are key amino acids. But doing all these exercises before that, try to understand what are your key amino acids, why you have to interact with that amino acids. It's very much important. Otherwise, doing this docking exercise, it's not of no use. Hope I answered your question. What can be done when mutation in a site? When you have mutations in site, you have to redock it. So consider replacing that amino acid. Again, do the docking exercise. Superimpose all those mutated binding sites and see for the same ligand whether there is a change in conformations for mutations, and whether there is change in energy, and what is the RMSD value for the same ligand in mutated proteins of the same pocket. What is the change in RMS? So this can be done visually by superimposing these binding pocket as well as the doctor ligands. Of course, I hope this will be the same ligand, then only you have a comparative analysis on mutated amino acids. Any other questions?
Hello, can you please again provide the link to your GD Bio webpage?
Okay. So that is script. You mean for the tutorial, madam?
For the tutorials and for COVID-19 data set and other
Okay.
information that you have compiled.
Okay. I will put it in my YouTube description so that you'll get all the links in there.
Okay, okay, fine, fine.
Any other questions? Oh, okay. There is a question. How to check the PDB quality? See, PDB quality, the crystallographic quality, we talk about resolution anyway. But otherwise, PDB quality on a biochemical perspective, we need to understand whether it is bioavailable, whether it is druggable. You have to read papers, and there are certain databases available to understand this mechanism. You have to check that. So, on analyzing all of them, only you can say how far the protein is going to be good. And in the RCSP website itself, at the bottom it tells you about the PDB quality. As you can see here, the PDB validation, it tells you about how far the PDB quality is. So you can go through this overall quality at a glance, which talks to you about the clash score, Ramachandran outliers, and side chain outliers. So this also tells you about the quality of the crystallographic PDB. Another question: how the glycerin linger involved in docking? Confirmation aspects. Sir, I really have not worked on it, but let me see how far the glycerin structure has significance. Sodium is there anyway, yeah. Okay, so I really do not know, sir. I need to check with that data. Or
Sir,
can you
sir,
explain the
sir,
question?
I have one doubt, sir. I am Suvaiti Namudan.
Okay, hi sir.
Sir, hi, sir. Sir, nice presentation, sir. Thank you, sir.
Thank you.
Sir, I have the 2000 small molecules.
Okay.
Only 2D structure. How to
Okay.
convert the 3D structure and the preparation, sir? Any online tools? I
Yes,
prepare
sir.
in.
Yes, sir. There is something, as I told you earlier, OpenBabel.
Open. Okay, okay, okay,
So in
okay.
OpenBabel, let me show you that either you can use a GUI, or you just go to the wiki page, where you will get a lot of information about the command line, where how to convert it from one let me tell you. So you have to give a command like OpenBabel
Okay,
minus
sir.
I, that is your input file, which is a 2D file,
Okay,
can
sir.
be mol2 or SDF. Then
Okay, sir.
space minus small o, again give as SDF,
Okay.
space minus capital O, and
Okay.
then give a file name, whatever
Okay.
3D file name, then
Okay.
space minus gen 3D,
Okay.
GEN 3D.
Okay.
That's it, enter. So all the 2D will be converted to 3D.
3D. Okay. Sir, okay, I convert the 3D.
Yeah.
Better one, AutoDock Vina is virtual screening, no, sir? Yes, one. That's one.
So AutoDock Vina can be used for virtual screen, but you need a software called as Pyrex. So
Pyrex.
Pyrex can be downloaded from this website, the
Okay.
scripts.edu. There's
Okay.
Pyrex here. So if you
Okay.
download that, you can load many molecules. But the problem with Pyrex, if you load more molecules, it will crash.
Crash. Sir. Okay. Okay.
Yeah.
AutoDock Vina, Vina, how much molecules runs?
No,
AutoDock
it
Vina.
can run any molecules. Vina
Any
has no
molecule.
issues. Only thing you
No
need
issue.
to have a script or an interface which handles virtual screening. That's
So
all.
AutoDock Vina is how to run, sir? It's the same AutoDock Like any script is available, sir. AutoDock Vina.
So I don't know. Did you watch my slide where I was telling you about? Just a minute. So did you see when I was showing you run AutoDock Vina here?
Oh, okay, okay.
So if you have missed it, the YouTube link, you can rewind and see. I have already covered how to run AutoDock Vina here.
Vina. Okay, 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 sir.
Yeah.
Okay, sir.
If you
Okay.
wanted
Thank
to
you, sir.
run, if you wanted to run in command line, that is also possible. What you have to
Possible.
do is, let me show you. So you have to give Vina.
Vina.
So minus H will give you what are the commands that you have to use to run Vina. Okay.
Oh, okay, sir.
Okay. Now what you have to do is Vina minus minus config space config dot txt. This will run AutoDock Vina.
Okay, okay,
So if
okay,
I
okay, sir.
run here, now you can see it is running now.
Okay, okay.
But ensure that your PDBQT of your protein receptor and your ligand in PDBQT both are saved in the same directory.
Same directory.
Yeah, now the results is ready. Now we will see what is there in the config.txt. So you can see this is what I have put in config.txt, so it will run.
Oh, okay, sir. Okay, sir.
Yeah,
Okay, sir. Okay, sir.
yeah,
Thank you, sir. Thank you, sir.
no
Thank you, sir.
problem. Thanks. Sir, I can see that now we will come to the drawback of AutoDock. So when you have metal ions or metal atoms, like I can see you have loaded silicon, parameterization is an issue. So when you try to generate interactions and parameterization is a bit difficult. It is not that you cannot do, you can do, and that's where we have to define the AutoDock parameter file and ligand parameter file. It's a bit advanced level where you have to play around with the text and then you have to mention the parameterization of your metal or let it be magnesium or, but it interacts. The question is whether it is reliable or not. So if your parameterization is better, AutoDock also can do it, but it's not a straightforward thing. Okay, another question is how far AutoDock is better than FlexX, which I already told you. When you're doing virtual screening, these other softwares would have made your life much easier to load the molecules much more conveniently rather than playing around with multiple softwares, multiple file conversions, multiple steps to do it. They try to minimize it. So it depends upon. So for example, for a PhD student or a student who is going to learn about docking, I always recommend to do AutoDock because he or she will learn what is at each step they have to do rather than doing a black boxing. I never said any softwares are bad, but don't think that just clicking button will get answers. We really need to go in depth and understand what really we are doing. So I would recommend for a person to start learning docking, the best tool is AutoDock or Doc6, which is freely available on the internet. And then when you have more demands, like as Sir was mentioning, as one Miss Anusha was mentioning, silicon atom having issues. Of course, then all these tools where they have much more advanced parameterization is going to help you with that. Otherwise, the parameterization is in AutoDock is possible, but you have to spend a lot of time on parameterization or creating the parameters for those atoms. Okay, any other, which one is user friendly? So let me tell you, for me, both are user friendly. It's about how you learn, how you spend time. For me, I'm been using AutoDock since 2004 or 5, I think. That time, this installation was not that easy like you do here. That time I have to install Sigwin, I have to install MGL tools, I have to install Python separately. All I have to install it separately that time. But now it's just a one software you install and everything works fine. So, so it's about how you learned it, how depth you know about it, and what all small things details you have gone deeper into it. That's where when I ask questions to a student in PDBQT, what is this Q and T? Most of the time they don't have an answer because they didn't go deeper into it. That's where we need to read the papers of Lamerican algorithm. We have to read papers of AutoDock. There they have clearly mentioned why we really require Trion or the charges and the torsions. So that is a level of depth of understanding that you have to do on any software. Any other questions? If there is no much questions, we can wind up the session, but let me know if you have any further questions. All the links I will put it in the YouTube web link, so it will be on the description of the YouTube web link.
Sir, how much extent we can depend on the reliability of docking result, especially for repurposing activity?
Yeah, so that is a very good question. I really like that. When you're doing a repurposing activity, you have to first validate the existing drug in a existing, which is reported target. First, you try to do a docking there, understand everything is fine there, and then bring that drug to your target, whatever you are trying to repurpose, and ensure that whether it is able to interact or with the key amino acids and the key atoms of the key amino acids. If it is not interacting with the key amino acid and still it is giving better score and better energy, what is the purpose of repurposing? So you need to have a better understanding of what should be my amino acid I should focus on interacting with. Let it be antagonist, let it be agonist. Whatever it is, you need to know the amino acid. So AutoDock, or let it be any software, you are the person who is going to make it much more reliable and reproducible. As you know, I always used to mention this word GIGO, garbage in, garbage out. So a person who is doing docking should know what you're really doing. If you're just simply doing, okay, let software take care of everything, then you get garbage. So you need to have a better understanding of what input you're defining to the software. So it's not about which software you're using. It's about what knowledge you have that is being inputted to the software. Let it be anything. So they are reliable if you have much better knowledge of the target and what you are trying to do. Any more questions?
Last one, sir.
Yes.
So for coders, it works with fine. It work fine with the Python interface, writing the commands and giving the instructions. But for persons who don't know the codings, which one is better?
As I told you, you really don't require coding here. Why do you require coding?
No,
Here,
sir, we have to write comments, right? That needs a practice and.
No, I didn't put any comments here. I said that is another option that is available.
Oh yes.
I never used any comments. So if you think that I hate comments, okay, don't do it. You can always do on the GUI side also.
Okay, sir.
Okay, so now I am going to present. Let us. I have few more questions that is there. So let me cover up those questions also. So just a minute. Okay, so okay, so I have like how to calculate binding energy. I would recommend you to read some reviews because normally we try to take all the energies like internal energy, electrostatic energy, and then try to come up with the binding. And so there's equations. So I didn't prepare any slides for that. So I would recommend you to refer to online sites, please. And the next question is, how to find a perfect docking? As I already shared a slide saying that a perfect docking means you need to know whether the scores are optimum, whether it is interacting with the right amino acids or not, whether there are no clashes, whether the torsions are acceptable or not, and also good significance or negative highest negative energy is available. That's where we try to call it as perfect docking. And keep in mind, docking is the initial phase on some objectives of the project, that to find heat identification. So that's where we wanted to understand which bonds are more stable. As you already know, hydrogen bonds are weaker bonds, right? So they are not the strong bonds anyway. So we have van der Waals interactions, Coulomb interactions possible, then electrostatic interactions, then hydrophobic is much more reliable. That's what I feel. And then that's where we have to give more importance. And then we have more questions please can we discuss about enzyme substrate docking? We wanted to inhibit that particular interaction, so we are trying to interact with some other ligand, but we need to have an understanding which amino acid is actually we have to inhibit. So that's why from the beginning of the session I'm telling you, you should have a thorough understanding about which amino acid I have to focus. So then you will get an answer for this question. I'm going a bit quicker, sorry, because it's already too late. Joining, I think it was posted earlier. Oh, sorry, these are all posted earlier. The manual suggestion that ADT is valid for correctly searching for conformational space for ligands with torsion DOF 10. Now, how should I restrict my DOF? Of course, you can modify your PDB-QT file to extend your torsion to 16, or when you are loading your input to ADT, there is an option to modify your torsions. So there is an option called, let me show it to you. Let me check. I don't know whether I will be able to show now. So you can, yeah, under Ligand, there is something called a torsion tree, and there you can set choose the torsions as well as set number of torsions. So there you can change from 10 to 16. So it is possible. Okay. Now, the next question, please explain about convergence criteria. Sometimes even after an overnight simulation while at redocking experiment, the software was not able to attain a result similar to the co-crystal structure with RMST less than 2. What to do? This is exactly what I told you. So when you were trying to do the docking parameters under docking, your initial geometry confirmation, do not make it as a random. Try to uncheck that random. By default, it will be random. Okay, uncheck it so that the center of the ligand will be defined as what you have defined in the grid box, so that you will try to achieve what you're looking at. So when you're doing a template or a co-crystal redocking, it's always better to uncheck the random thing, so that the starting will be always your initial geometry. What would you suggest the best DPF parameter are for exhaustive search? As I told you already, genetic runs, please make it to 1,000 or 10,000. That will do an exhaustive search, but it takes more time. How to integrate AutoDock Vina result with AutoDock? Is it preferred, or just AutoDock Vina is enough for a concise docking experiment? As I told you already, once you redock with your co-crystal ligand and confirmed the parameterization for your grid box, what you have used, the sender, the grid points, whatever you have used is validated, then that is enough for your config file to AutoDock to work nicely. And AutoDock Vina is much more accurate than AutoDock. I know you will be surprised, but there are papers to prove that. So I used MGL Tools and AutoDock to just validate my co-crystal ligands and the grid points. That's all. If you do not have a co-crystal ligand, still you have to do it to validate it. And then do your AutoDock Vina exercise for virtual screening. There are scripts available, many of them. How different is AutoDock Vina results from AutoDock? As I told you, AutoDock Vina results are much better than AutoDock, with a very minimal difference, with a 0.2 or 0.4 RMSD values, but not tremendously high. But on historically, with many analysis, AutoDock Vina is much more having better results than AutoDock. Kindly provide the, okay, so one is in script, SCRIBD, script.com/bdbio is where you can find the tutorial for AutoDock
MGL Tools with GUI access as well as command line. Then slideshare.net/GiriBio will get you access to all the open slides and presentations. And github.com/GiriBio will give you access to the COVID datasets and many more other machine learning based Python scripts. What is protein feature view of protein? Sorry, I really didn't get this question. Can you please, if you're on the line, can you unmute yourself and explain what is this question? Because all your questions are anonymous. If you want to reveal who you are, you can explain this question. I really didn't understand. Sorry. Will the AutoDock results be enough to publish in a good Scopus Index journal? Of course, yes. I have published AutoDock results in 14 impact factor journals. If you think impact factor is what you look at a good thing. For me, a good publication is when my publications get cited in different journals. That's where I say, okay, wherever I publish is a good journal. Of course, that relates to impact factor, but for me, that my personal view is if my paper is getting cited a lot, not self-citation, what I mean, then that refers to me that is a good journal. Not always good impact factors refers to good journal always, okay. I don't deny that, but I don't want to generalize it. So it's always that no journals are asking you whether you are used a paid software or not. Nothing. Your discussion should be more strong, whether you use paid or free. Nobody going to ask. I am a reviewer for ACS journals, Elsevier journals. I have get a lot of papers on many tools being used. I never asked about paid or free. Many have used free tools. Only thing I ask them, justify it, whether it's reproducible, whether it's reliable. Whatever you have explained, your discussion part is not enough. You wrote what software gave. Come up with your ideas. Come up with your interpretation. If it is a bioinformatics or biotechnology paper, give me a biochemical perspective on your docking results. That is what I expect from you. Don't think that AutoDock is a very good software, but you have to use it in the right way. But there are limitations, as I already told you. So keeping that in mind, if your problem statement suffice with all the features available in AutoDock, go for it. How to get interactions via ADT Vina? This is a good question. I forgot to mention that. Of course, I try to go much detail, but I missed this. In AutoDock, of course, you get interactions from AutoDock tools. For AutoDock Vina, you use ligplot to get the interaction, or you can use PyMol or Chimera to get the interactions. If you want to go a bit more, if you want to go a bit more advanced, there are free visualizers available for Windows and Linux. You can download them and you can load your PDB and your PDB Qt file. Of course, they are open there. And then you will see that they are engulfed like a docking. And then there are scripts there to define the binding site and ligand interactions. But keep in mind, there are plenty of other interactions also will come there. So you have to choose which one you want to consider and not. After docking, hydrogen bond within, of course, hydrogen bond within ligand is important, but keep in mind they are weak bonds. They are not strong bonds. So considering that, you can, because salt bridges will be there, water bridges will be there with hydrogen bonds, they take place. They are important. Any other questions? So if you have any further questions, of course, you can unmute yourself. Otherwise, I can end the session. There is 2 more sessions coming up. On coming Thursday, we have a start drop demo, which is completely on the role of ADME and toxicity on drug candidate selection. So after doing docking, you get the hits. After getting the hits, how do we make selection? Because hit identification is like potency test. We just look at the inhibition, how far it tightly binds to the target. That's all. But that's not enough for you to call it as a lead molecule or a drug-like molecule. So the next step is that what to do with pharmacokinetics and what is the role of a pro-drug strategy.
So those things we will be looking on April 23rd at 4 p.m. And there will be an expert talk discussion at Saturday. It will be an expert will be invited. I will let you know who is the person and his expertise in the coming days. So thank you all for your time and patience. And
Hello.
if you, yeah,
This is Praveen again, sir.
yeah, Praveen, tell me.
Actually, I'm new to this AutoDock software, and usually I civil and discovery student, sir.
Okay.
Is this software is compatible with Windows 10 and any other specification is needed, like
In the beginning of this
graphics cards?
session, I already told you it is compatible with everything except Mac OS Catalina version. So otherwise, all operating system it works. No issues.
There is no graphics card needed like that.
No, no, not required. Not
Thank you.
required. Yeah. Anything else? Okay, so thank you all so much. I know it's took a bit lengthier than expected. I expected only 2 hours, but I hope it was useful. I hope I covered much in detail, and you will be able to do more exercises. And if you have any questions, I'm happy to answer. You can either contact me through WhatsApp or through email. I'll be putting my email also into the YouTube description link. That's the only way that I can communicate with you, because I do not collect your phone numbers or email address, which I do not want, and so that for privacy issue. So I will be better posting it there so that you can contact me. Thank you so much, and have a good night, and stay safe. Bye.